All right, today we're going to go through Chapter 10, which is the nervous system. This is going to be broken up into multiple recordings just since it's such a long chapter. I want to try to keep the recordings kind of short. So first we'll just talk about the nervous system in general. It consists of the brain, spinal cord, and all of the nerves. It is a very, very complex system, so hopefully this lecture is going to help to fine-tune tune you into what you need to know regarding this chapter. So what does the nervous system do? It constantly receives signals about changes within the body as well as the external environment. It then processes the info, decides what action needs to occur, and sends chemicals and electrical signals to the cells, instructing them how to respond. We're going to talk about all of those different things. A couple of different divisions of the nervous system. We have the central nervous system, which consists of the brain and the spinal cord. And we have the peripheral nervous system, which consists of the nerve network, pretty much everything else outside of the brain and the spinal cord. This chart here helps to divide it down a little bit more. So, of course, we have the central nervous system, which is the brain and the spinal cord. It's really the peripheral nervous system that has a lot more pieces to it. So we have a sensory division or a motor division. You need to understand that the sensory division carries signals from nerve endings to the central nervous system. And then the motor division transmits information from the central nervous system to the rest of the body. So the sensory division takes stimuli to the central nervous system. Then the motor neurons or the motor division brings that information back to the rest of the body and tells the body what to do with it. Within the sensory division, we have the somatic sensory and the visceral sensory. So the somatic sensory carries signals from skin, bones, joints, and muscles. And the visceral sensory carries signals from the viscera of the heart, lungs, stomach, and the bladder. So more kind of internal. Then in the motor division, we have the somatic motor and the autonomic motor. So the somatic allows voluntary movements of the skeletal system. And then the autonomic provides these automatic divisions such as blood pressure, heart rate. It plays a lot with our internal organs. That system is then broken down into the sympathetic division, which arouses the body for action. So kind of that fight or flight, if you guys have ever heard someone mention that. And the parasympathetic division, which is the calming effect. So they kind of play off of each other. And we'll talk about that towards the end of this chapter. So the neuralgia is one type of cell within the nervous system. We also have neurons. We briefly talked about those within the last couple of weeks. Just to help you understand a little bit, glia, so neuralgia, that G-L-I-A at the end of it, really means glue. So that kind of tells you a little bit the purpose. The purpose of these supportive cells of the nervous system really are to hold the neurons together. There's about 50 glial cells for each neuron, and there is about 5 different types of these neuralgia. The first 4 types there are in the central nervous system, and the last one is the only one in the peripheral nervous system. So the oligodendrocytes form the myelin sheath within the central nervous system, and the Schwann cells form the myelin sheath in the peripheral nervous system. The ependymal cells secrete cerebrospinal fluid, or CSF. The micralgia perform phagocytosis, so it can actually get rid of some of the bacteria. And astrocytes form the blood-brain barrier. On that note, how do these astrocytes form the blood-brain barrier? They actually wrap around the capillaries and they protect the brain from foreign substances. So we need to have this semipermeable membrane because it allows small molecules like oxygen, carbon dioxide, and water to diffuse across to the brain, but blocks larger molecules. This helps protect the brain from foreign substances. It also prevents most medications from reaching brain tissue. So it's kind of a protective mechanism that our body designed to protect our brain. And although neurons don't undergo mitosis, so they do not go through cellular division, glial cells do and are often the cause of brain tumors. So usually when you hear of somebody having brain tumors, it's a glioma that they have, and it's actually a neuralgial cell that's causing the tumor, not an actual neuron. Neurons are the other part of the nervous system, and they really handle communication. They are the ones that send those signals back and forth between the central nervous system and the peripheral nervous system. So the sensory neurons, like I said before, they detect stimuli, things like touch, pressure, heat, cold, or chemicals, and they transmit information about the stimuli to the central nervous system. Interneurons, which are only found in the central nervous system, connect the incoming sensory pathways with the outgoing motor pathways. So they're kind of in between these two different neurons. And then the motor neurons, they relay the messages from the brain to the muscle or the gland cells. So they are the messenger. Here's the structure of the neuron. Neurons have multiple different types of shapes and sizes, but they have three basic parts, a cell body and two extensions called an axon and a dendrite. And we did mention this also a couple weeks back. So the cell body, also known as the soma, is the control center and contains the nucleus. The dendrites receive signals from other neurons and conduct the information to the cell body. So they receive the stimuli. Some neurons have only one dendrite and others have thousands of dendrites. So it's really dependent on where the neuron is. The axon then carries the nerve signal away from the body of the cell and on usually to the next neuron. Cells only have one axon, and the length of the axon ranges from a few millimeters up to a meter. The axons of many neurons are encased in a myelin sheath. We mentioned just a few slides ago that there were cells, glial cells that make the myelin sheath in the peripheral nervous system and the central nervous system. And what that does actually insulates the axon. We're going to talk more about that coming up. You can see it here. It's basically what happens is it allows the impulse to go faster through the neuron so that we can have these kind of split-second reactions. There are some little gaps here in between them, and those are called the nodes of Ranvier. And then all the way at the end of the axon, we get these axon terminals, and right at the end is what is called a synaptic knob. And those are the ones that really contain the neurotransmitters. And again, we mentioned that a little bit before, but we're going to talk in more detail about it. And that's what sends the signal on to the next neuron. So here's the myelin sheath. This is what it kind of looks like if you take a cross section of it. And the peripheral nervous system versus the central nervous system looks very similar. There is myelin sheaths in both areas. There's different cells that are responsible though, and we talked about that already. So we have the Schwann cells for the peripheral nervous system, and we have the oligodendrocytes for the central nervous system that make this myelin sheath. The other difference is that in the peripheral nervous system, these nerve cells are covered with neurolemma. It's this outside kind of protective barrier, and it actually allows these cells to regenerate. However, in the central nervous system, they do not have that protective function. So central nervous system neurons cannot regenerate, which means that if there's some sort of damage to the spinal cord, it is not really fixable. And that's why people become paraplegics or quadriplegics. Most of the neurons are covered with these axons, but some are really not. And the ones that aren't are the ones that are not really dependent upon speed or getting the message back and forth very quickly. So, an example of this would be neurons within the stomach. We need to tell the stomach to secrete stomach acid, but it's not something that needs to happen super quickly. So those neurons are not covered in myelin sheath. So let's talk about impulse conduction, because this is what really happens within these neurons. So it's really caused by an electrical current. This results from a flow of charged particles from one point to another. So really in the body, whenever ions with opposite electrical charges are separated by a membrane, usually the cellular membrane, the potential exists for them to move toward one another, and this is called membrane potential. So that's opposite electrical charges separated by a membrane. Polarization, on the other hand, is a membrane that exhibits membrane potential, an excess of positive ions on one side of the membrane and excess of negative ions on the other side. This is what we consider polarized. So they would say the membrane is polarized when it has membrane potential. So let's walk through the steps of impulse conduction. The first step is resting potential. So when a neuron is not conducting an electrical signal, The interior has a negative electrical charge and the exterior has a positive charge. The outside of the cell is rich with sodium ions, whereas the inside contains an abundance of potassium ions. And because of this, we get, some well, not because of this, but the membrane has permeability, right? So a certain amount of sodium and potassium ions leak across the membrane. However, the sodium and potassium pump constantly works to restore the ions to the appropriate side. And we learned about that sodium-potassium pump back a few sections. This state of being inactive and polarized is called the resting potential. So the neuron is really resting, but it has the potential to react. So it's ready to react. It's ready to create a stimuli or an electrical current. The next step that happens is depolarization. So a stimulus, such as chemicals, heat, or mechanical pressure, causes channels on the neuron's membrane to open, and sodium from the outside of the membrane rushes into the cell. The addition of the positively charged ion changes the charge of a region of the cell's interior from negative to positive. So we have all of these positives rushing inside. As the membrane becomes more positive, it's said to depolarize. So in polarization, when the cell is polarized, we have this kind of ability for it to react. We have lots of positives on the outside, lots of negative on the inside. What happens here is that the membrane allows all of those sodium ions to rush in and changes the inside to positive. This actually starts to create this electrical current. So in step 3, we have this action potential, and we have the depolarization kind of going above what they call a threshold level. So enough sodium ions rush in that it causes adjacent channels to open, allowing more sodium ions to enter, and this creates this action potential, which really means that the neuron has become active as it conducts an impulse along the axon. Another term for this would be called nerve impulse. So stimuli creates this nerve impulse or this action potential. The action potential continues down the axon as one segment stimulates the segment next to it. So it continues all the way down the axon. After that happens, repolarization happens. So the sudden influx of sodium triggers other channels to open, and this allows potassium to flow out of the cell. Soon after potassium begins to exit, the sodium channels shut to prevent any more sodium from flowing into the cell. This repolarizes the cell. However, sodium and potassium are now flip-flopped, with the outside containing more potassium and the inside containing more sodium. So we have our electrical charges back correctly, but we have the ions on the wrong side. So during then the refractory period or step 5, as long as these sodium and potassium ions are on the wrong side of the membrane, the neurons won't respond to a new stimuli, even though the membrane is polarized. This is called the refractory period. So we have to kind of wait for the sodium-potassium pump to return the sodium to the outside and the potassium to the inside. When this is complete, the nerve is again polarized and in resting potential until it receives another stimuli. So I'm going to show you the animation on this just to review what we just talked about. And hopefully it will help to get you to understand it a little bit better. Let me just find the right one here.
To relay messages to organs and tissues throughout the body, nerves must initiate and then transmit signals from one neuron to the next neuron at lightning speed. Signal transmission occurs through an electrical current. When a neuron is not conducting an electrical signal, its interior has a negative electrical charge, whereas the charge on the outside is positive. The outside of the cell is rich with sodium ions, whereas the inside contains an abundance of potassium ions. The interior of the cell also contains large, negatively charged proteins and nucleic acids. These additional particles give the cell's interior its overall negative charge. Because of the membrane's permeability, a certain amount of sodium and potassium ions leak across the membrane. However, the sodium-potassium pump constantly works to restore the ions to the appropriate side. This state of being inactive and polarized is called resting potential. The neuron is resting, but it has the potential to react if a stimulus comes along. When a stimulus comes along, channels on the resting neuron's membrane open, and the sodium from outside the membrane rushes into the cell. The addition of all these positively charged ions changes the charge of a region of the cell's interior from negative to positive. As the membrane becomes more positive, it is said to depolarize. If the stimulus is strong enough, adjacent channels also open, allowing even more sodium to flood the cell's interior. This creates an action potential, meaning that the neuron has become active as it conducts an impulse along the axon. The sudden influx of sodium triggers the opening of other channels to allow potassium to flow out of the cell. Soon after potassium begins to exit, the sodium channels shut to prevent any more sodium from flowing into the cell. This repolarizes the cell. However, the outside now contains more potassium, and the inside contains more sodium. Although the membrane is polarized, the neuron will not respond to a new stimulus as long as the sodium and potassium are on the wrong sides of the membrane. The sodium-potassium pump works to return sodium to the outside and potassium to the inside. This is known as the refractory period. When the sodium-potassium pump restores sodium ions to the outside of the cell and potassium ions to the inside of the cell, the nerve is again polarized. It remains in resting potential until it receives another stimulus.
All right, so hopefully that just kind of reviews it again for you one more time. And just to be clear, this is impulse conduction in unmyelinated fibers, so there is no myelin sheath on those fibers. What we're going to talk about next, I'm going to show you the animation and how it's slightly different when we're talking about fibers that have this myelin sheath on them. So let's watch that. And then you'll be able to see the difference. Come on now.
The axons of many neurons are encased in a myelin sheath. Consisting mostly of lipid, myelin acts to insulate the axon. The thick layer of myelin blocks the free movement of ions across the cell membrane. The only place ion exchange can occur is at the nodes of Ranvier, the evenly spaced gaps in myelin. Electrical changes occur at the nodes of Ranvier, creating an action potential. The current flows under the myelin sheath to the next node, where it triggers another action potential. This process continues as the signal moves down the axon. Because the action potentials occur only at the nodes, the impulse seems to leap from node to node. This type of signal conduction is called saltatory conduction.
So you see how that is different than when the sheaths don't have any myelin. So it actually jumps from one node of Ranvier to the other because it has this protection, and that actually allows that impulse to go faster. So the last part I want to talk about here, just in a general sense of the nervous system, kind of finishing up our conversation on the neurons, is the synapses. So this is where the action potential comes in and it reaches the synaptic knob, so this kind of end part of the axon here. And the membrane depolarizes, the ion channels open, calcium ions enter the cell. So right, that's happening as this action potential comes down. And at the end here, it changes a little bit because we get the calcium instead of the sodium. So the infusion of the calcium causes vesicles to release their store of a neurotransmitter into the synapse. There's tons of different neurotransmitters, so here we're just talking about it in a general sense. The neurotransmitter binds to receptors on the postsynaptic membrane. So here these little vesicles, you know, come to the edge of the synaptic knob here, and they're excreted into this little space here, and then they flow across and they attach to these little areas here that send the action potential onto the next axon. So this is the axon. The axon sends it into the next dendrite, right? The dendrite receives signals, sends those to the cell body of the neuron. The neuron then continues the action potential down the axon if necessary. So there's like I said, lots of different neurotransmitters. And the specific neurotransmitter determines whether the impulse continues or whether it stops. So sometimes we want this stimuli to actually stop. And then after the receptor releases the neurotransmitter, it is reabsorbed by the synaptic knobs and recycled or destroyed. So we can reuse any that are left over here. Let's watch an animation quickly on this.
Nerve impulses usually travel through several different neurons before reaching their target organ or tissue. For this to happen, the impulse must have some way of transferring from the axon of one neuron to the dendrite of another neuron. The area where this occurs is called a synapse. Most synapses do not touch. Instead, a chemical called a neurotransmitter bridges a very narrow gap called the synaptic cleft to carry the message from the first neuron, called the presynaptic neuron, to the next, called the postsynaptic neuron. The process begins when an action potential reaches a synaptic knob and the membrane depolarizes. This causes ion channels to open, which allows calcium ions to enter the cell. The infusion of calcium causes vesicles to release their store of a neurotransmitter, such as acetylcholine, into the synapse. Once released, the neurotransmitter binds to receptors on the postsynaptic membrane. This causes sodium channels to open and sodium diffuses into the cell. The addition of the positively charged cells causes the membrane to depolarize, and the impulse continues. The receptor then releases the neurotransmitter, after which it is reabsorbed by the synaptic knobs and recycled or destroyed by enzymes.
So this is how communication from one neuron goes to the next neuron. Because we have so many neurons throughout our body, we need this communication to kind of continue. So this is how it happens in between the ends.